This is a recorded lecture for Chapter 8. This is looking at bacterial genetics. This is Microbiology 230. I am Dr. McGraw. This is really the chapter, the continuation of Chapter 7, where we looked at basically the blueprint of life from DNA to protein. This is really what happens within the bacterial world, and that genetics occur within there. So, with a glimpse of history, if you will, Barbara McClintock lived from 1902 to 1992, observed that kernel colors of corn were not inherited in a predictable manner. He concluded that segments of the DNA moved in and out of the genes or involved in color and destroyed the function of the genes. So published results in 1950 met with skepticism. DNA was believed to be stable. The idea established by 1970s, and she was awarded the Nobel Prize in 1983. And they termed this the transposons, or jumping genes. Hard to say fast, really. Jumping genes is the word. So let's look at antibiotic resistance, if you will. Staphylococcus aureus, or Staph aureus. It's a gram-positive cocci, commonly called staph, frequently the cause of skin and wound infections, and since the 1970s has been treated with penicillin-like antibiotics up to the point of methicillin. In fact, it's been treated so much, if you will, that we've developed these multiple strains called methicillin-resistant Staph aureus. So in 2004, where 60% of Staph aureus strains from hospitalized patients were resistant to methicillin, creating this MRSA, methicillin-resistant Staph aureus. We project that 2.3 million healthy people in the U.S. harbor this MRSA, if you will. Healthcare-associated MRSA, if you will, resistant to other antibiotics, including vancomycin, and vancomycin is considered the drug of last resort. We now have 3 cases that I know of in the research literature that shows that we have wounds, and we have wounds that were infected with Staph aureus, MRSA, which has no antibiological treatment. So basically, we've reached the end of our antibiotic bullets, if you will, and we have nowhere to go at this point. Now, genetic change of bacteria organisms can adapt to changing environments. Through natural selection, it favors those with greater fitness. Bacteria adjust to new circumstances, and there's a regulation of gene expression, which we've already seen in Chapter 7. And we'll talk about genetic change in this chapter. So bacteria are excellent systems for genetic studies because they have rapid growth. They occur in large numbers through the logarithmic growth. There's more known about E. coli genetics than almost any other type because it's the most common, and it also is a part of the microbiota in our gut. A change in organism's DNA alters the genotype, and so the sequence of nucleotides in the DNA is then changed, and bacteria are haploid, so only one copy, there's no backup involved, so they're not a diploid, it's not a diploid chromosome. They may change observable characteristics or phenotype. Remember, the genotype is what all the genes that are maybe seen or not seen in their behavior or structure. Phenotype is what you see, and so they're also influenced by environmental conditions. So it gives a look at that. Mutation and horizontal gene transfer. What you're seeing is you'll see this organism which is going to go through a normal mitosis, and what happens is there's spontaneous mutation in the one genome. So what happens is they have these organisms, they'll go through the next stage, if you will, the next mitosis, if you will, and so now this group, if you will, are going to be different than the first group. So what happens is we have the first group which doesn't have this change, and the other group which does, and there can be a plasmid transferred from one to the other to make them so they have that same ability. So if it's good or bad. Now, mutation change could be fatal, it could be prevented from bacteria, it could be lots of things depending on what's going on. And mutations can change the organism's phenotype easily because there's a deletion of genes for tryptophan biosynthesis yields a mutant that only grows if tryptophan is supplied in the environment. So growth factors require the mutant term an auxotroph. Oxo means increase, troph means nourishment, so it needs a specialized nourishment. A prototroph doesn't require growth factors, so proto means first. Now, geneticists compare mutants to the wild type, and mutants are, if you will, are the typical phenotype of strains isolated from nature. For example, wild type E. coli strain is a prototroph. The strain is designated by 3-letter abbreviations. For example, TRP cannot make tryptophan minus. Streptomycin resistant is designated tryptophan R, or STR. So that's just ways we identify each of those different strains. Now, we also have spontaneous mutations, which are caused by just normal processes. They occur randomly at infrequent characteristic rates. There's no occurrence, no given occurrence that given every third generation or whatever, but they have a predictability. So mutation rates, probability of mutation, each cell division, typically between 10 to the 4th and 10 to the 12th for a given gene. So 10 to the 4th means 4 times 10, if you will, 4 zeros, if you will. So that would be 3 zeros, and then 4 zeros would be 10,000, as it were. And then we have we can move on to 10 to the 12th. And so anyway, mutations are passed to progeny. Occasionally change back the original state. You can have some reversion. Large populations may contain mutants. For example, cells in a colony may have developed these mutations. And that's one of the problems with growing bacteria in plates, is that you may take your specimen from a mutated colony to do your investigation, and it may not be representative of the rest of the bacteria in that group on that plate. So it's kind of one of the things you have to deal with. So environment selects cells that grow under its conditions. For example, antibiotics are selected for resistant bacteria present. The environment does not contain mutations necessarily. Single mutations are rare, 2 are even rarer, and so physicians may use 2 antibiotics to reduce resistance and change a product mutation rate for each one. So the chance of that happening is based on each one combined. So base substitution is the most common. Incorrect nucleotide is incorporated during DNA synthesis. Point mutation is change of a single base pair. You can see that in this graphic. Base substitution leads to 3 possible outcomes: a silent mutation, which is a wild type amino acid; a misused or missense mutation, which is a different amino acid. Resulting protein may only partially function. Term leaky is a term we use if it's in a cell wall, like an ag or NAD. A nonsense mutation, which means that the protein doesn't even have any sense at all. That doesn't have any function at all, and it may well be a fatal outcome. Now, base substitution of mutation inactivates gene is termed a null or knockout mutation. A silent mutation sometimes is used to indicate a mutation that does not alter the protein function, but may change it in some way. Base substitution is more common in aerobic environments. The reactive oxygen species produced from O2, you can oxidize the nucleobase guanine, and DNA polymerase often mispairs it with adenine. So that occurs more often than other mutations. Great proofreading function of DNA polymerase. If you look here, I'll let you download that. By the way, again, you have to download the PowerPoint, and then you have to run the slide show version of that. And then if you get to this point, you click on there, and you can play these various videos. Now, deletion or addition of nucleotides and our spontaneous mutations as we go forward. The impact depends on the number of nucleotides, obviously. And there are 3 pairs that change this one codon. So one amino acid, more or less, and that's impacts depends on the location where the protein's at. 1 or 2 pairs yields what's called a frame shift mutation. And that's where different sets of codons are translated. Often results in premature stop codon, shortened non-functional protein, or knockout mutation is the most common result. Again, addition and deletion mutations, great video. Take a look at that. Spontaneous mutations. We talked about transposons before. They're jumping genes. They can move from one location to another. The process is called transposition. Transposons will be the jumping genes that perform the transposition. The gene is insertionally inactivated, and the function is destroyed. So most transposons have transcriptional terminators, blocks the expression of downstream genes in the operon. So basically, it often results in a death or a fatal flaw at the end. There's a simple transposon video. You can look at that in your on the PowerPoint. This looks at the mechanism of transposition, again, in the PowerPoint. Now, classic studies carried out by Barbara McClintock showed the observed color variation in corn kernels resulted from transposons moving into and out of genes controlling the pigment synthesis. Now, induced mutations are a little different. They result from outside influences. For agents that are agent that induces changes called a mutagen. For instance, X-rays or chemicals or heat or extreme cold. Geneticists may use mutagens to increase the mutation rate. There are 2 types that we use, chemical and radiation. And there's the list of chemical and radiation choices, the action and the result. I'll let you look at those. Now, Chemical mutagens may cause base substitution or frame shift mutations. And some chemicals will modify the nucleobases even. They change the base pairing properties, so increase the chances of incorrect nucleotide incorporation. So for instance, nitrous oxide converts cytosine to uracil, or cytosine to uracil, excuse me. And the base pairs with adenine instead of the guanine, which creates that difference. Here's a look at mutation by base substitution. I'll let you look at this video as I described before. Some chemicals modify the nucleobases, and they are called alkylating agents. They add alkyl groups into the nucleobases. The nitroguanidine adds methyl group to guanine. The base pairs with thiamine, and so you get a pretty significant change if you look at the methylguanine of the template strands with the thiamine instead of the cytosine, and you see the difference. Now, base analogs resemble nucleobases, so they have different hydrogen bonding properties. They can be mistakenly incorporated by DNA polymerase. The 5-bromouracil resembles the thiamine. Often base pairs with guanine, and so 2-amino purine resembles the adenine, often pairs with cytosine. So you can see that how that can even become a mutator in its own right. We have intercalating agents which cause a frame shift mutation. They're flat molecules that intercalate or insert between adjacent base pairs in the DNA strand. It basically pushes the nucleotides apart of the original DNA and produces space, and obviously it causes errors during replication. If it's in a template strand, a base pair added to the synthesized strand. So if in a strand being synthesized, the base pair is deleted, often results in premature stop codon. Ethidium bromide, or ethidium bromide, excuse me, is common intercalating agent. It's likely carcinogenic. So ethidium bromide will cause a carcinogenic response or cancer-like response. Chloroquine, used to treat malaria, is another example of this intercalating agent. The transposition, we talked about transposition can be used to generate mutations. Transposon inserts in the cell's genome and generally inactivates the gene to which it inserts. So usually it has a fatal flaw at the end. This looks at transposon, the shifting segments of the genome. Great video. Again, I strongly suggest you take a look at it. Radiation is another element that causes an induced mutation. There's a couple of techniques that do that, a couple of types of radiation. There's ultraviolet radiation, and there's X-rays. Ultraviolet radiation forms these thymine dimers. In the covalent bonds between the adjacent thymines, it cannot fit in the double helix. It distorts the molecule. As you see here, it creates this abnormal sort of event. Replication transcription solves that distortion. The cell will usually die if the damage is not repaired. Mutation results in the cell's SOS repair mechanism. So you'll see a repair technique that will take this out, put something else in. The X-rays cause a single and double strand breaks in the DNA. The double strand breaks are often produce lethal deletions, and X-rays can alter the nucleobases directly. This is another video. Let's take a look at how that happens, or how that works. Second one is direct repair, how that DNA molecules, the thiamine dimers can be repaired. Now, we look at repair of damaged DNA. There's an enormous amount of spontaneous and mutagen-induced damage to DNA. If not repaired, it can lead to cell death or cancer in animals. For example, in humans, 2 genes associated with breast cancer code for DNA repair enzymes. Mutations either result in 80% probability of breast cancer. So that's pretty important to know and pretty medically pertinent. Mutations are rare. Alterations in DNA are generally repaired before being passed on to the progeny, the next generation. And so several different DNA repair mechanisms are used. This looks at addition and deletion mutations. Again, another great video. This is the repair of damaged DNA. It looks at spontaneous and mutagen-induced. I'll let you read through this. I won't go through it in this video, in this lecture, because it would take us forever. But I'll let you take a look at that, and it'll be self-explanatory. Now, the repair of errors in nucleotide incorporation during replication, DNA polymerase sometimes incorporates the wrong nucleotide. And so there's a mispairing which slightly, or I'll say distorts the DNA helices in a minor manner. And it's recognized by certain enzymes. So the mutation is prevented by repairing before the DNA replication occurs. These enzymes will see that distortion and make the correction. There are 2 mechanisms: proofreading and mismatch repair. And proofreading by DNA polymerase, it verifies the accuracy of the DNA. It can back up or excise a nucleotide, a dysfunctional nucleotide. It can incorporate the correct nucleotide. It's very efficient, but it's not flawless. There are mistakes that are made in this process. This is a methyl-directed mismatch repair video. Mismatch repair. It fixes errors missed by the DNA polymerase. The enzyme cuts the sugar phosphate backbone. Another enzyme degrades the short region of DNA strand. Methylation of the DNA indicates the template strand. And the methylation takes time, so newly synthesized strand is unmethylated. DNA polymerase and DNA ligase make those repairs. So that's a much more complicated, much more sophisticated mechanism to create that change based on that mutation. Now, repair of modified nucleobases in DNA, they lead to base substitution. So glycosylase removes the oxidative nucleobase, and another enzyme cuts the DNA at this site. DNA polymerase that removes the short section, synthesizes the replacement, and the DNA ligase seals that gap with hopefully the proper nucleotides. Now, repair of the thymine dimers, there are several methods to repair damage from UV light. There's photoreactivation, that is light repair. The enzyme uses energy from the light. It breaks the covalent bonds of the thymine dimer that we talked about, and it's only found in bacteria, not in humans. Excision repair, that's a dark repair. That means the enzyme removes the damage. DNA polymerase and DNA ligase repair the area and hopefully put in the correct nucleotides. This is a nucleotide excision repair video, good thing to look at. So there are several methods we talk about repairing from UV light. There's SOS repair, and it's the last ditch repair. And SOS means, if you remember SOS in the Morse code, means danger or danger. Last ditch repair mechanism. It is induced following extensive DNA damage. Photoreactivation and excision repair were unable to correct the problem. The DNA and RNA polymerases stall at the unrepaired site, so several dozen genes in the SOS system are activated. It includes a DNA polymerase that synthesizes even in extensive damaged regions. It has no proofreadability, so errors can be made and results in SOS mutagenesis. So basically, in the end, you create a mutant, if you will, based on this mechanism. It may be better than it was before, but it still has that single problem. Now, mutants are rare and difficult to isolate. There are two main approaches: direct selection. The cells are inoculated into medium that supports growth of mutant, not the parent. For example, antibiotic-resistant mutants are exposed to the antibiotic. Indirect selection isolates the autotroph from the prototroph, or auxotroph, excuse me, from the prototroph parent strain. So more difficult since parents will grow in any medium in which auxotroph can grow, and you have a replicable plating. So this looks at the techniques, if you will, that we just talked about. Now, here's the process we take. If we take this, we have a plate of bacterial colonies. It's pressed into the surface of a sterile velvet. The master plate with microbial colonies, the nutrient auger, is nutrient auger, the sterile velvet, and the colonies will be imprinted on the velvet. We have cells adhering to the velvet are then transferred to the sterile medium, resulting in stacked replicas of the original plate, and we start to see if there's a nutrient auger where all colonies grow, or a glucose salt auger in which auxotrophs do not grow. So it'll tell us the difference between those two. You tell which one to switch. Now, mutant selection, penicillin enrichment of mutants sometimes is used. It increases the proportion of auxotrophs in a broth culture. So penicillin kills growing cells, like prototrophs and auxotrophs survive. So it kills the prototrophs, but the auxotrophs survive. So penicillinase has been added, and the cells are plated in a rich medium, and you can identify the auxotrophs from the prototrophs. Now, we can also do a screening for possible carcinogens. Carcinogens cause many cancers. Most are mutations or mutagens. The animal tests are pretty expensive, time-consuming, and PETA doesn't really like that very much. Pretty inhumane. So mutagens increase low frequency of spontaneous reversions. We have what's called an Ames test, which measures the effect of chemical on a reversion rate of histidine-requiring salmonella auxotroph. It uses direct selection. It's mutagenic. The reverse rate increases relative to control. But for instance, rat liver extract is added, since non-carcinogenic chemicals are often converted to carcinogens by animal enzymes. So additional tests on mutagenic chemicals are used to determine if it's carcinogenic. So that's one of the tests we use, the Ames test. This is takes the process of that. We have a control plate, then we have the test plate. In the control plate, the bacteria requiring histidine for growth, the glucose salts and agar, and they lack histidine. The liquid without a suspected mutagen is added, and the spontaneous HIS pluses are reverted. We see that a few colonies form. In this case, the bacteria requiring histidine for growth are added. glucose salts, the agar, the liquid containing the suspected mutagen is added, and we see the many colonies form here. We see the few colonies on this side with the HIS negative side, histidine negative. Now, horizontal gene transfer is a mechanism of genetic changes also to be considered. Microorganisms are commonly acquired genes from other cells, which is called horizontal gene transfer. They can demonstrate recombinance with autotrophs. Auxotrophs, I'm sorry, I keep saying autotrophs because obviously it's a name for bacteria. We categorize bacteria. Auxotrophs, it combines two strains. For example, histidine negative, tryptophan negative, leuco negative, and THR negative. And spontaneous mutants are unlikely, so colonies that can grow on glucose salts medium most likely acquire genes from another strain. Genes are naturally transferred by three mechanisms: transformation, transduction, conjugation. Transformation is the naked DNA is taken up by the bacteria. Transduction is bacterial DNA is transferred by viruses. Or conjugation, where DNA is transferred during cell-to-cell contact via a sexual pili, and that looks at those different mechanisms there. This looks at bacterial conjugation. Again, I'll let you look at that if when you download the PowerPoint. This looks at transduction generalized. Again, download the PowerPoint. Bacterial transformation. Again, download the PowerPoint. Sounds like a broken record, doesn't it? So DNA replication only if replicon, so it has original replication. Plasmids, chromosomes are included. DNA fragments enter the chromosome via homologous recombination, and only if the sequence is similar region of the recipient's genome. So what you're seeing here is that you're seeing a non-integrated DNA fragment that's included, and it just is basically will be included in some of the progeny. In this case, it's not part of the homologous with the recombination. So if it is combined, what happens is it's combined with the original DNA, and then it's replicated, so all the progeny will have this DNA segment, as it were. This looks at integration of excision of a plasmid. Again, I'll let you look at that on your video. Now, DNA-mediated transformation. Naked DNA is not within the cell or virus. The cells released when lysed. Additional, for instance, cells lysed and this DNA is released. Addition of DNA prevents the transformation. The demonstration of transformation in pneumococcal is only encapsulated cells are pathogenic. So look at the original injection. We have these live encapsulated cells of pneumococci, streptococci, and the mouse dies. If we take off the capsules, we have living non-encapsulated cells. We have no effect. So the heat kills the encapsulated cells. There's still no effect. If you kill the encapsulated cells, but we add the other cells to the mixture, what will happen is all the cells will become encapsulated again, and then the mouse dies. So these encapsulated cells are able to retrain the non-encapsulated cells how to make capsules. Now, transformation, the recipient cell must be competent, must take up regardless of origin, except only from closely related bacteria DNA sequences. So, in other words, sometimes the DNA, the recipient cell has to be similar species or genus, if you will, to the original cell. Sometimes not. The process is tightly regulated. For instance, Bacillus subtilis has two component regulatory systems. It recognizes low nitrogen and carbon and high concentration of bacteria, which is the quorum sensing. Only a fraction of the population becomes competent, and thus 1 out of 3 you'll see here is non-transferred cells. You'll see that. You'll see the 2 are transferred. 1 out of 3 will die in that process or have a fatal flaw. Now, transduction is transfer of genes by bacteriophages. So basically, specialized transduction from specific genes. We saw in Chapter 13. There's generalized transduction of 8 genes in the donor cell. It's a rare error during phage assembly, and the transfer of the DNA to the new host occurs. And we've seen this process before, where basically what happens is the DNA is incorporated. It's been through mitosis, it's incorporated into the progeny, and at a certain point, a trigger is reached where all the progeny, at whatever stage that is, will then the virons will then be released from that progeny and at one time. Conjugation of DNA is transferred between bacterial cells, requires contact between donor and recipient cells, and then they have to have conjugated plasmids direct their own transfer, so they have replicons. The F plasmid, for instance, fertility of the E. coli is the most studied. Other plasmids include resistance to some antibiotics, and they spread resistance easily. And they obviously go out the sexual pili. This is a good conjugation transfer of chemical DNA. Again, a video to look at through the downloaded PowerPoint. Now, F plasma of E. coli, the F plus cells have it, the F do not, and so it encodes the proteins, including F pelus. It's a sex pelus that brings cells in contact. The enzyme cuts the plasmid. A single strand is transferred. Complementary strands are synthesized, and both cells are now F plus when it's done. So it's transferred the ability, whatever that is, that the plasmid carries. Now, chromosomal DNA transfer is less common. It involves what's called HFR cells, high frequency of recombination. The F plasmid is integrated into the chromosome via homologous recombination. The process is reversible, and the F plasmid results when small pieces of chromosome is removed from the F plasmid DNA. So F is the replicon in this case, the term we use. Now, HFR cell produces the F pelus. The transfer begins with genes on one side of the origin of transfer of plasmid in the chromosome. Part of the chromosome is transferred to the recipient cells. The chromosome usually breaks before the complete transfer, so the full transfer would take about 100 minutes. The recipient cells remain F minus in the sense the incomplete F plasma is transferred. So even though there's been a transfer, it's not been enough of the information to allow it to gain the information that was meant to be said. There's also a mobile gene pool. Genomics reveals surprising variation in gene pool in even a given species. So perhaps 75% of E. coli genes are found in all strains, and they're termed the core genome of the species Escherichia coli. The remaining make up the mobile gene pool, or the variable gene pool, or the transfer gene pool. All those terms are used. Plasmid, transposons, genomic islands, or phage DNA all fall in this mobile gene pool. And this looks at those different groups and what they are: transposons, plasmids, genomic islands, and phage DNA. We've talked about those before, but that's a nice table for sort of bringing it all together. Plasmids are found in many bacteria archaea. Some are even eukaryotic cells. They're usually they're double-strand DNA with the origin of the replication. They generally are non-essential. Cells survive without their loss, and there are a few too many genes. They have a low copy number, or a few per cell, and they are a high copy number, maybe is up to 500. Most have a narrow host gene, host range, so they only work with a specific gene species, and some have broad host range, which include gram-negative as well as gram-negative as well as gram-positive. So these plasmids can be sent from a gram-negative cocci, or gram-positive cocci to a gram-negative bacteria, a gram-negative rod, as it were. Very interesting how that can happen. We also have resistance plasmids, those R plasmids we talked about before. They help promote resistance to antibacterial medications, or heavy metals, mercury, arsenic, and the compounds are found in hospital environments. There's often two parts: the R genes and the RTF, or resistant transfer factor. These are the codes for conjugation. They often have a broad host range, which means they can affect one can be sent from one genus to several other genuses. And the normal microbiota can even transfer to pathogens. So you can, for instance, we used to be that Staph aureus was a pathogenic version of the most invasive version of Staph, and Staph epidermidis was pretty much non-invasive. But over the years, through mutations and through transfer of plasmids, some Staph epidermidis has become pathogenic. And so now we now refer to Staph aureus as coagulase positive. We refer to Staph epidermidis as coagulase negative. We can't refer to them as one being pathogenic, one not being pathogenic anymore, when at one time that was so. Now, transposons provide a mechanism for moving the DNA. They can move into other replicons in the same cell. Its simplest is the insertion sequence. It encodes only transposase enzyme in inverted repeats. The composite transposons include one or more genes and integrate via this non-homologous recombination we talked about. So this is the technique you see here. Now, transposons yield vancomycin resistance. So the Staphylococcus aureus strain, the Staph aureus. The patient was infected with Staph aureus, which was susceptible to vancomycin, but also had vancomycin-resistant strain of intercocci fecalis cells. So the E. fecalis transferred via pili the resistance ability to the Staph aureus. And the transposon jumped to the plasma of the Staph aureus, and thus the Staph aureus became vancomycin resistant. Now, the interesting thing, I want to insert this here, the interesting piece here is that we are looking now to see if we can reverse that effect. So we can reverse technology, and create bacteria that are no longer resistant. So we take a bacteria that are infecting somebody, insert a mechanism to reverse their ability to be resistant, and use that as part of the treatment. Bacteria can conjugate with plants. They're natural genetic engineering. For instance, Agrobacterium tumefaciens causes a crown gall, which is a growth. The different properties produces opine or plant hormones. A piece of tumor-inducing plasmid called tDNA, transfer DNA is transferred to the plant. It's incorporated in the plant chromosome via this non-homologous combination, and you see the change in these growth, these galls, as it were. Now we have genomic islands, which are large DNA segments of the genome. They originated from other species. The nucleobase composition very differently from the genome. And the GC pair, the guanine-cytosine base pair, ratio characteristics for each species change. They may provide different characteristics. For instance, utilization of how energy sources are used, tolerance to acidic environments, development of symbiosis with the host, or ability to cause diseases. Pathogenic islands are referred to. So these are techniques that are used. So what we've done here is we've looked at the bacterial genetics, which is the next step from Chapter 7, and we're done with this lecture. Thank you very much. Have a good day.